Okay, so welcome everyone to the third Pages North American Reps ECN show and tell, a special show and tell, a special event featuring a young researcher in our field. In this case, we're going to be hearing today from Pablo Martínez Sosa. He is a PhD student here at University of Arizona, working in the lab of Jessica Tierney. It's a paleoclimate lab. He's a specialist in organic geochemical proxies from lakes. The focus of his current thesis work is on these enigmatic biomarkers called BR-GDGTs, which are made by bacteria and used in all sorts of paleoclimate reconstructions. But they're kind of a rapidly developing field with a lot of discoveries, really, reshaping the field every month coming out. Pablo has already published several first author papers in this field, which is an impressive feat for a PhD student. Some of his past work has used mesocosm and microcosm applications to understand the ecology of the microbes that make BR-GDGTs. And most recently, he published a globally relevant calibration for applying this proxy to understand paleotemperatures of lakes and other parameters. Before his paleoclimate work, he conducted research in immunology at the National Autonomous University of Mexico, so he is quite the polymath. Though anyone who has followed his social media will know he's very passionate about GDGTs. Today, Pablo will discuss his everyday life in the lab and his research, starting with a presentation and opening up into a Q&A. If you want to ask questions, you may do so in the chat box, or turn on your mic at the end to ask questions when we say it is okay to do so. Until then, we request everyone mute their mics to allow Pablo to take our undivided attention. Now, let's hand it over to Pablo to hear from him about his work. Go ahead, Pablo.
Hi, everyone. Thanks to the Pages representatives for inviting me. I'm very excited to share my work here. As Dan mentioned, I'll just start with a little showcase of overview of what we've been working on here in the organic geochemistry lab at U of A, and then I'll give you a tour of the lab, and hopefully you'll enjoy the tour. So yeah, thanks again. Well, as Dan mentioned, the focus of my research are biomarkers, and in particular, microbial biomarkers. The ones I study more strongly are branch glycerol, the glycerol tetraeters, or branch GDGTs for short. And this is a family of 15 different structures. You can see kind of the overview here of the structure of these molecules. The main difference between these 15 different species is the number of methyl groups, from 4 to 6. The 5 and 6 methyl groups can be the 5th or 6th methyl group can be on the carbon 5 or 6. We have these other isomers, so that also increases the number of GDGTs. And they can have a cyclopentane rings from 0 to 2. So all these different 15 different variants vary on these different parameters. And branch GDGTs are really interesting because they're distributed worldwide. We find them in soils, peatlands, lakes, rivers, etc. And they're actually really resilient molecules. We found them up to the Cretaceous, I think. So they're really resilient to degradation. Now, based on the structure that we have, because these are actually really interesting molecules for a biological sense, they kind of look like a mix between what we would expect from bacteria and archaea lipids, which are like completely different organisms. So it's really, it was a puzzle for a while. The key difference that why people believe they're being produced by bacteria is the chirality of the glycerol groups, which are classically bacterial. So people believe these are bacteria in origin. And I say believe because we really don't know what organism is producing them. We found one bacteria is shown here, which is Edapobacter aggregans. And it has been shown that these organisms produce one of the 15 species. So we still have 14 different orphan molecules. And so it's a very interesting thing. The one thing we do know is that when we compare the distribution of these 15 different molecules with environmental factors, there's a strong influence. Here I'm showing a graph where we use an index that shows the degree of methylation of these different species with temperature for soils and lake sediments. And you can see that it has a really nice linear correlation. So we do know that the environment affects the branch GDGTs. We just don't know how the environment is affecting the GDGTs, who is producing them, and how they are being produced. So right now they're pretty much working like a black box, which is super interesting for a research point of view, but it can also be very frustrating at times. So overall, in our lab, what we've been focusing is these like big three questions that I put here. What can we learn from the source organisms, considering that we don't know who they are? What environmental parameters affect the branch GDGT distribution? And do all the environments have the same response? I told you they're found in soils, peatland, lakes, rivers, but are they behaving the same way? So to do this, we use an approach which allowed us to both avoid having to culture the organisms and preserve some of the environmental parameters from the natural sources. So we collected water from a lake nearby and generated some microcosms. These were like 1 liter bottles that we incubated at different temperatures or different pHs. And again, what this allows us is to control a single environmental parameter, but also preserve some of the water chemistry that could be important for the microorganisms. So we want to learn here some details about the organism itself and how they are responding to and which environmental parameters they respond to. We were fortunate to be able to pair this lipid analysis, which is what I'll be focusing today, with some genetic sequencing, which it's also a pretty interesting result and aspect for this. But overall, what this part of my project showed is that temperature is the strongest influence on branch GDGTs for lakes, at least. And again, I showed this index versus temperature, and you can see that we were able to recreate this linear relationship between temperature and the branch GDGTs. Looking a little bit at the microbial abundance, because we don't know who's producing the branch GDGTs. And we don't even know if a single organism is producing all 15 or several organisms are just like changing the microbial community and producing these different molecules. So we were able to sequence these microcosms and do some analysis on the microbial community. And what we found, and what I'm sharing here, is that the microbial communities strongly change with this temperature, which is not surprising, but we were actually able to show that some variation on the branch GDGTs change varies with like certain lipids. So we think that could, and other groups have also suggested that it's more of a microbial community change. What drives this response to the environment. Again, at least for lakes, temperature is the strongest influence. Now that we have this idea of what parameters are influencing the GDGTs, we decided to, take a step back and look globally how this is working out. Because, we were looking at a single lake in Arizona. So how does that compare to other lakes globally? And for that, we were able to generate 500 lake, 227 global calibration from lakes all over the world. Here's the distribution. It's not only our samples, we also collected some samples from incorporated data sets from other researchers. But as you can see, we have a decent coverage of the world. Again, we try to look at the environmental parameters that could be influencing this. And we were actually, again, found that temperature is the strongest influence for lakes, but secondarily, pH and the conductivity of the lake seem to also exert some influence, though not as strong as the temperature. We generated this calibration that ideally we could apply to other unknown samples. And as I'm showing here, we compared that with a reconstruction on Maine, and our results were comparable with instrumental data, which was really, we were pretty excited about. An interesting factor is that you can see here that I'm not plotting this graph against a mean annual air temperature, but rather mean temperature of months above freezing, which is what we found to be the best driver, even on temperature standard. Finally, again, I mentioned we don't really know if the branch GDGTs are behaving the same way in all the different environments. So the last section that we're starting to work on is, again, taking a further step back and looking at marine samples, lake samples, rivers, soils, and peatlands. And here the question we're trying to answer is, looking at the GDGTs, and here we're expanding further from the branch GDGTs to some archaeal molecules, can we find like traces using machine learning to then, like characteristic patterns, I guess, to then take samples whose the positional environment is unknown, and using GDGTs be like, oh, this is actually a peatland, or this was a lake, or this was a transition between peats and lakes. So that's also a very interesting section that we're starting to work on now. And yeah, I skipped all the experimental work, which is, I guess, what we're here for. And yeah, with that, I'll move to the tour section. And I guess you can type questions. I'm not sure if you can unmute yourself, but yeah, if any questions you may want to ask. Thanks for listening. I'm just gonna mute here. Can you see me? Okay. So I'll be giving you the tour, the multi-step process from the samples we have to what we get. And I would say the picture I show you where I'm collecting water is probably one of the only instances where our lab group did some field work. We actually just like get samples from some collections, which you can see here. Everyone has their own samples stored here. And based on the research, we have everything from like rocks to sediment samples here. Yeah, but we can, for the microcosm, for example, I used water, which needed to be filtered. To actually start extractions though, we need to get rid of all the water. And we use this machine here, the freeze dryer. So all the samples are stored in this chamber, and with a vacuum and low temperature, we're able to like freeze dry them. The samples are then grinded. We have some mortars over there. And what we want to do is then extract all the organic matter that we can get from these samples. I'm just gonna show you here. This is the accelerated solvent extraction machine we have, the ACE. And this is what will allow us to do the organic extraction. What we need to do for that to happen is to use these cells that are here. So once we have our sediments grinded or processed as the sample may require, we need to combine them with diatomaceous earth. And we're gonna be using these cells for the ACE machine. As you can see here, we have a 3-part cell that we will assemble, fill with the diatomaceous earth, plus the sample. Here we have some glass fiber filters. And these cells are placed on the revolver here. And the best description of this machine is basically an expensive Nespresso machine. So the cells will go here in the revolver and then get here into the oven and the machine. They're heated, and heated solvent, in this case we're using dichloromethane with methanol, is like pressured into the cell and then is placed here in this other revolver where each sample is placed. We use these cells here. And I think there's a question here. Yeah, cross-contamination, it's a pretty big issue. I was gonna show you here. All the material we have is glass, and everything is ash first. Afterwards, every of the glass material is always cleaned with methanol and then ECM. The other question, which I lost. Okay, about the preservation. Yeah, as I show, we mostly store the samples in the fridge. So I guess mostly what we don't want is like any volatiles that could, evaporate. Yeah, so continuing here, we get the sample here in these cells, which are filled with solvent. And we'll start a process that you'll see a pattern here. At this stage in the process, we have a vial full of this solvent, organic solvent with our material that we want to analyze. So what we proceed to do is use this machine, this turbo bath, and it's basically a water bath that has nitrogen flow. So we would place the samples here and just like blow down all the solvent that we have here. So we get only the organic samples that we're interested at the bottom of the vial. Now, what we can do now is and I hope it's not too loud now. Yeah, so the samples are here and we blew them down to be just the organic. We now can use different solvents. We mostly use CCM isopropanol to, dissolve everything in a volume that's manageable. And we use these glass pipettes, which I'm not sure you can see here. And we transfer all the some to these 4 ml vials. And again, this is what I say, there's a pattern here. Again, we have all the organics in this solvent, and this is placed on the FlexiVac, which is a heated plate, which has nitrogen. We blow it, all the solvent down, get the organic sample here. And now we're gonna use the silica gel columns. Here we just want to separate all the organics based on certain characteristics, polarity or something like that, depends on the sample we're using. So I'm not sure if we have this with glass wool and fill it with silica gel. And now the samples are transferred. In my case, what we used was hexane, which is a very apolar solvent. Dissolve all the organics here, transfer to the column, let all what's soluble in hexane to transfer to these solar vials. Always 3 times rinses. And then we use hexane ethyl acetate, which is a more polar solvent. Again, dissolve the sample in that solvent and transfer to the column. So we're able to separate the GDGTs, for example, which are polar in these hexane ethyl acetate. But depending on what we need, the column can change. We can use samples just of silica gel or different mixtures of the solid phase, or the solvents will also allow us to separate different molecules. Once again, you may be guessing that the samples that are dissolved on the different fractions will go to the FlexiVac, be blown down. It's a lot of waiting for a solvent to get dissolved. And then they're transferred to these tiny vials 2 ml vials, which is what we can use to load the Hplc, which is what we use for GDGTs. So here's our Hplc mass spectrometer. It's called nicknamed Saguaro. You can see here the samples are placed here, and then we can have a flow of the solvent to the samples. There's a column here, and then we'll go to the mass spectrometer, Saguaro. And yeah, we can then measure the peaks and quantify the organic molecules based on the areas. Okay. And yeah, that's pretty much the process we use for the branch GDGTs. We can use different processes, especially on the first section, depends on the sample, what we're using. What we'll do, for example, extracting liquids, we analyze mezcals at some point, and that uses a completely different extraction process since it's liquid, so we don't use the A's. But yeah, other than that, I guess we can move on to questions if that's okay.
And I could read questions to you, Pablo, if you'd like.
Yeah,
up to you. All right. So we have a question from Zhao asking, I wonder if, number one, do you find that other environmental factors, for example, light, nutrients, may affect BR-GDGTs? And number two, is it possible that microbial produced GDGTs could behave differently in a very close distance, less than a kilometer, and similar elevation due to microenvironment condition?
Yeah, that's a great question. Based on our results, at least in lake sediments, the temperature response is really, really strong. I think in our principal component analysis, it showed that about 60% of the variance is explained just by temperature, with pH and conductivity maybe. The PC that best correlates with those parameters is about 20%. Obviously, that leaves another like 20% of variance that we cannot explain. So it's possible that those other factors are affecting, but should probably be like lower influence, or maybe going like more locally. You can, yeah, actually when you focus on a single place, you actually start seeing other factors. For example, there's a paper from Ioana Stefanescu when they actually see a depth effect on the lakes. So yeah, there's definitely influence. Looking more globally, I guess, there's temperature is the strongest for lakes. For sediments, for soils, for example, pH is the strongest influence. And that leads to the second part of your question. For example, lake sediments and soils on the surrounding are really, really different. So I can see how that would affect. Again, how much microenvironments based on other factors would affect, I'm not sure. Again, since I work mostly on lakes, it's assumed that it's mostly like everything is mixed. So I'm not sure how strong that effect is on soils.
All right. We got another question from Ricardo Santos. Have you tried to do the TLE extraction manually in order to assess the differences between ACE results? Because he was wondering the possible temperature effect of ACE on the compounds.
Yeah, we've done that 2 times now. We use the classic acid extraction versus ACE extraction, because yeah, there's this question of whether we could get different results. For the most part, we don't see, we get slightly different yields, but the distribution of GDGTs is preserved in both extraction methods. So it's faster, the ACE, and if I remember correctly, it was actually a slightly better yield in the ACE for us at least, for our application. Because going, for example, for the microcosms, we actually use acid extraction. But yeah, otherwise it's been pretty similar.
Interesting. While we wait for other questions, I have one. How did you get involved in this in paleoclimate more broadly, and then also specifically in this topic of Lake BRGDGTs?
Yeah, so actually, my undergrad, it wasn't quite immunology. It's called basic biomedical research. It's a very unique degree on Unam. And what I did there was move from more biomedical to biogeo area. So I work with Dr. Rafael Navarro looking for evidence of life on Mars, which was this like good combo both. And that led me to try to find projects where you would be mixing these again microorganisms with the environment in geological samples, and that's how I ended up working with just UNA.
Gotcha. And where do you see, you're kind of at you're in a rapidly developing field with a lot of, you know, pretty formative discoveries happening all the time. Where do you see your field going in over the next few years?
That's a good question. I think what my experience says, or what I've seen, is that while branch GGTs were found several years back, it has mostly been used by organic geochemists and paleoclimatologists without much regard for the biological part. So I think we're now catching up, and hopefully in a couple of years we can get a more complete, again, going back to the picture I showed you, the diagram, all those unknowns hopefully will get completed. Probably just, you know, we'll turn to a tool. I don't think GDGTs are the best tool. They're just a fun tool.
Yeah. We got another question from Tamara Fletcher. How biased are GDGT temperature estimates to seasonal variation versus annual averages?
Right. What we found is that there's not quite a seasonal variation, but rather, as I mentioned, the parameter that we found to be the most useful is mean temperature of months above freezing. So what we calculate is the mean temperature of all the months that didn't go down below zero. So it's not quite seasonal, but there's definitely this argument that either the organisms go dormant during the cold season, or they just get, there's this idea of getting shielded by ice cover on the lakes from the actual temperature. So that can disrupt the calibration. And you can see that when you compare temperate lakes with more tropical lakes, where you don't see that much of a bias.
Oh, she says, right on. I didn't understand what you were saying earlier. Yeah, that's interesting about the phenology of, do you think that's the seasonal phenology, the bacteria is affecting when they are recording the environment accurately? Do they go into hibernation during the winter months?
I think there's some evidence for that. Again, since we don't really know the organisms, it's hard to tell. I would imagine that's the case, or at least the community is changing slower, or they just get biased. But yeah, it could be that it's a more physical phenomenon.
And it sounds like you're interested in that, the ecology of the not the animals, the organisms. That I have the same thing happens in other paleoclimate fields. Like there's people like me that want to understand the clams, and then other people who just see the clams as a mean to an end. So it's interesting to see that tension. Do we have any questions from other folks? One new message. All right, let's see. Ricardo asks, does lake water stratification also yield a shield effect as in the ice cover?
Oh, that's actually a pretty good question, because yeah, and I guess this comes back to another question that we have. One environmental factor that we've seen does affect, not quite on the distribution, at least in our experiments, but rather how much GDGTs you get is oxygen concentration. Other research has shown that the source organisms may be located on the transitional zone between the oxic and anoxic. So they're not quite either, but that region gets like preferential production. So there's definitely some, I think some research in Lake Chala has shown that you can see how the stratification of the lake affects the production of branching keys. So, I mean, for temperature, it could be the case, but also it's really interesting that the oxycline affects the production of the branching keys.
There are other people who study GDGTs in oceanic settings, correct?
Yeah, mostly for oceans, they're isoprenoidal GDGTs, which are archaeal in origin. Those are known to be archaeal. So while the structure are similar, they're pretty different in origin, I guess. But you do find some branching keys, and that's what we're trying to combine both for the more machine learning general research.
Gotcha. Asif asks, how impressive are GDGTs in examining lake anoxic, euxinic, or eutrophic conditions? Is there any linkage with them? I guess you'd be asking how effective are they for the investigating anoxia, I guess.
Yeah, honestly, I'm not sure if like that has like done more like systematically. Like if we, yeah, I know there's been shown that even the community changes when people have analyzed the oxic and anoxic part of lake, but like a more general like calibration like research, I'm not aware that exists for lakes of this.
Out of curiosity, which lake did you go to in Arizona?
It's Kennedy Lake. It's a tiny artificial lake that's mostly used for fishing.
Oh,
Yeah,
gotcha.
we're not famous for lakes, I guess.
Yeah, I've been to a few up in the White Mountains and stuff, and they're up there, but yeah, they're not exact. We're not the land of lakes here, so it's just funny to be a lake doing. It's kind of like me growing clams in Arizona. It's not a likely place to do it. But yeah, do we have any other questions from folks? Well, we're at about 40 minutes, so that makes sense. But do you have any closing remarks, Pablo, before we close off this session?
Branch GDGTs are pretty cool. You should consider using them.
Yeah, I like that. I like it that you're an evangelist for this proxy, and anyone who is interested in following Pablo more can check out his Twitter account, which is, I believe, Terrible Lagarto.
Yep.
And of course, he has several recent manuscripts in this field. But did anyone have any oral questions? Nope. I think we're okay. All right. So with that, I will close off this successful show and tell. We'll be having more starting in the next couple months. And thank you for coming. And thank you, Pablo, for talking to us. Yeah, it was really interesting seeing your POV of your research process.
Yeah, thanks for the invite. It's pretty fun.
Awesome. All right.
Thanks everyone for attending too. It's really nice to see the turnout.
Yep, and stay tuned for future events coming up in the next few months.